Greetings, my name is Deja Crossman, and this is my presentation on the regulation of glycolysis and gluconeogenesis. Glycolysis is a central metabolic pathway that occurs in the cytosol of all cells. In this process, glucose is broken down into two molecules of pyruvate. During this breakdown, the pathway generates energy in the form of ATP and reducing power in the form of ADH. An important feature of glycolysis is that it can function under both anaerobic conditions and aerobic conditions. In contrast, gluconeogenesis is essentially the process of synthesizing glucose rather than breaking it down. This pathway primarily occurs in the liver and, in a lesser extent, in the kidneys. Glyconeogenesis becomes especially important during fasting, starvation, or prolonged exercise, when blood glucose levels begin to drop. During these states, the body produces glucose from non-carbohydrate sources, such as lactase and certain amino acids, to maintain a stable supply of glucose for tissues that depend on it, especially the brain and red blood cells. Because glycolysis and gluconeogenesis perform opposite metabolic functions, the body tightly controls them through a process called reciprocal regulation. This ensures that when glycolysis is active, gluconeogenesis is inhibited, and when gluconeogenesis is active, glycolysis slows down. This coordination prevents a futile cycle in which glucose could be simultaneously broken down and synthesized, which in turn would just waste ATP and cellular energy. One important characteristic of glycolysis is that it does not require oxygen, which means it can function under both aerobic and anaerobic conditions. Because of this, glycolysis is especially important for cells that either lack mitochondria or frequent experiences of low oxygen levels. For example, red blood cells rely completely on glycolysis to produce ATP since they do not contain mitochondria. The pathway can be divided into two major phases: the energy investment phase and the energy payoff phase. During the energy investment phase, the cell uses two molecules of ATP to phosphorylate glucose and its intermediates. The phosphorylation process adds phosphate groups to the glucose molecule, which destabilizes it and prepares it for subsequent breakdown. Although this step requires an initial input of energy, it is necessary to make the glucose molecule more reactive and easier to split later in the pathway. After the molecule is prepared, glycolysis enters the energy payoff phase. In this stage, the six carbon molecule that originated from glucose has already been split into two 3-carbon molecules. Each of these molecules continue through a series of reactions that produce energy. During this phase, 4 ATP molecules are generated through a mechanism known as substrate level phosphorylation. In addition, 2 molecules of AD plus are reduced to ADH, which can later contribute to further energy production in the electron transport chain when oxygen is available. We can calculate the total energy produced. We must subtract the 2 ATP molecules during the investment phase from the 4 ATP produced in the payoff phase. This results in a net gain of 2 ATP molecules per glucose molecule. Although Along with these 2 ATP molecules, glycolysis also produces 2 NADH molecules and two molecules of pyruvate. These products can then enter other metabolic pathways such as the citric acid cycle under aerobic conditions or fermentation under anaerobic conditions, depending on the cell's environment and energy needs. Glycolysis contains 10 enzymatic reactions, but only 3 of these steps are considered the 3 major regulatory points of the pathway. These steps are essentially irreversible under physiological conditions, which makes them ideal control points for regulating the overall rate of glycolysis. The first regulatory step is catalyzed by hexokinesis or gluconeogenesis in liver cells. These enzymes convert glucose into glucose 6 phosphate by adding a phosphate group using ATP. This reaction is important because phosphorylation traps glucose inside the cells. Once glucose is converted into glucose 6 phosphate, it cannot easily cross the cell membrane and is therefore committed to cellular metabolism. Hexokinesis is found in most tissues and has a high affinity for glucose, while gluconeogenesis is primarily present in the liver and pancreatic cells and functions when glucose concentrations are high, such as after a meal. The second and most important regulatory step is catalyzed by phosphofructokinesis 1, commonly abbreviated as PFK1. This enzyme converts fructose 6 phosphate into fructose 1 or 6 biphosphate. This reaction is often described as the committed step of glycolysis, meaning that once this reaction occurs, the glucose molecule is fully committed to being broken down for energy. Because of this, PFK1 serves as the primary control point of the pathway and responds strongly to signals reflecting the energy status of the cell. The third regulatory step occurs at the end of glycolysis and is catalyzed by pyruvate kinesis. This enzyme converts phosphoenolpivalate or PEP into pyruvate while generating ATP through substrate-level phosphorylation. Since this is the final step of glycolysis, it also plays an important role in controlling how quickly the pathway proceeds. All 3 of these enzymes are regulated through mechanisms such as allelogenic regulation and covalent modification, including phosphorylation and dephosphorylation. These regulatory processes allow cells to rapidly increase or decrease glycolic activity depending on metabolic conditions, such as energy demand, nutrient availability, and hormonal signals. This type regulation ensures that glycolysis operates efficiently and responds appropriately to the energy needed by the cell. 1 is considered the most important regulatory enzyme of glycolysis because it catalyzes the committed step of the pathway. The it is important to note that the activity of this enzyme is highly controlled by molecules that reflect the energy status of the cell. First, we look at the negative regulators. When the cell already has abundant energy, the concentration of ATP is high. ATP acts as the allosteric inhibitor of PFK1. Importantly, ATP does not only bind to the enzyme's active site where the reaction occurs, it also binds to separate regulatory sites. When ATP binds to the regulatory site, it changes the enzyme's shape and reduces its affinity for the substrate fructose 6 phosphate. As a result, glycolysis slows down because the cell does not need to produce additional ATP. Another inhibitor is citrate, a molecule produced in the citric acid cycle. High citrate levels signal that the cell already has sufficient metabolic intermediates and energy, further suppressing PFK1 activity. In contrast, positive regulators activate PFK1. When the cell needs more energy, the ATP is being consumed rapidly, and levels of AMP and ADP increases. These molecules bind to the enzyme and reverse the inhibitory effects of ATP. This activation stimulates glycolysis so that more ATP can be generated to meet cellular energy demands. One of the most powerful activators of PFK1 is fructose 2 or 6 biphosphate. This molecule greatly increases the enzyme's activity and can even overcome the inhibitory effects of high ATP concentrations. It is also important because its levels are regulated by hormones such as insulin and glucogen. In this way, it acts as a metabolic signal that coordinates glycolysis with the body's overall nutritional and hormonal state. Gluconeogenesis is the metabolic pathway responsible for the synthesis of glucose from non-carbohydrate precursors. This process occurs primarily in the liver, with the kidneys also contributing, especially during prolonged fasts. The main purpose of gluconeogenesis is to maintain stable blood glucose levels when dietary glucose is unavailable, such as during fasting, starvation, or extended periods of exercise. This is essential because several tissues, including the brain and red blood cells, rely heavily on glucose as a primary energy source. Several different substrates can serve as starting materials for gluconeogenesis. One important source is lactate, which is produced during anaerobic metabolism in tissues such as skeletal muscle and red blood cells. Lactate is transported to the liver where it's converted back into glucose through the Cori cycle. This recycling system allows the body to recover carbon molecules from lactate and reuse them for glucose production. Another key substrate is glycosyl, which is released during the breakdown of triglycerides in adipose tissue. Glycerol contains three carbon atoms and can be converted into intermediates of gluconeogenesis, allowing it to serve as the building block for glucose synthesis. Amino acids also play a major role in glyconeogenesis. Many amino acids can be converted into metabolic intermediates that feed into the pathway. One particularly important is adenine, which is transported from muscles to the liver through the glucose adenine cycle. In this cycle, muscles release adenine during protein breakdown, and the liver converts it into glucose that can be returned to the bloodstream. Unlike glycolysis, which produces ATP, glyconeogenesis requires a significant input of energy. In fact, synthesizing one molecule of glucose requires the equivalent of six high-energy phosphate bonds typically provided by ATP and GTP. Although this process is energy intensive, it is crucial for maintaining blood glucose levels and ensuring that vital tissues continue to receive the adequate supply of energy during periods where dietary glucose is limited. Gluconeogenesis cannot simply run glycolysis in reverse because several steps in glycolysis are irreversible. Instead, the body uses special enzymes to bypass these steps. These enzymes represent the key control points of gluconeogenesis pathway. The first bypass reaction is catalyzed by pyruvate carbio, car, carbolase, carboxylase. This enzyme converts pyruvate into oxaloacetate within the mitochondria. The reaction requires ATP and the cofactor biotin, which helps transfer carbon dioxide during the reaction. Pyruvate carboxylase is activated by acetyl CoA, a signal that the cell has sufficient energy and metabolic intermediates available to support glucose synthesis. The next step involves PEPCK. This enzyme converts the oxaloacetate into phosphoenolpyruvate, or PEP, using GTP as an energy source. Unlike many metabolic enzymes that are controlled by intermediate cellular conditions, the PEPCK is also strongly regulated at the level of gene expression. Hormones such as glucagon can increase its production during fasting, while insulin decreases its expression after eating. Another major regulatory step in the gluconeogenesis is catalyzed by fructose 1. This enzyme removes the phosphate group from fructose 1 to produce fructose 6 phosphate. It acts as a key control point in the gluconeogenesis and functions in direct opposites to the phosphofructokinesis 1 in glycolysis. This reciprocal relationship helps to ensure that glycolysis and gluconeogenesis are not highly active at the same time. Finally, glucose 6 phosphate catalyzes the last step of gluconeogenesis. This enzyme is located in the endoplasmic reticulum of the liver and kidney cells. It converts glucose 6 phosphate into free glucose, which can be released into the bloodstream. This step is essential for maintaining blood glucose levels during the fasting because it allows newly synthesized glucose to be distributed to other tissues throughout the body. Regulation is a key mechanism that prevents glycolysis and gluconeogenesis from occurring at high rates simultaneously. This coordination ensures that the cell does not waste energy by running two opposite pathways at the same time. One of the most important molecules controlling this balance is fructose 2. This molecule acts as a powerful metabolic signal. It strongly activates phosphofructokinesis 1, the rate-limiting enzyme of glycolysis, while at the same time inhibiting fructose 1, the key regulatory enzyme of gluconeogenesis. Because it has opposite effects on these enzymes, fructose 2 helps determine which pathway is active in the cell. The levels of fructose 2 are controlled by special biofunctional enzymes, which is called the PFK2. This enzyme has two different catalytic activities. One domain functions as a kinesis that synthesizes fructose 2, while the other acts as a phosphatase that breaks it down. Which activity dominates depends on whether the enzyme is phosphorylated or dephosphorylated. Hormones regulate this process. In the fed state, the hormone insulin signals that blood glucose levels are high. Insulin causes dephosphorylation of the bifunctional enzyme activating the kinesis portion. This increases the concentration of fructose 2, which stimulates glycolysis so that cells can use glucose for energy or storage. During fasting, the hormone glucagon becomes dominant. Glucagon activates protein kinesis A, which phosphorylates the bifunctioning enzyme. This enzyme activates the phosphorylase portion, lowering levels of fructose 2. As a result, glycolysis is inhibited and gluconeogenesis is stimulated, allowing the liver to produce glucose and maintain blood glucose levels for the rest of the body. Hormonal regulation plays a critical role in coordinating glucose metabolism across the entire body. The two primary hormones involved are insulin and glucagon, which respond to changes in blood glucose levels and signal tissues to either store or produce glucose. Insulin is released by the pancreas when the blood glucose levels rise after a meal. Its main function is to promote the uptake and utilization of glucose by tissues. Insulin activates protein phosphatase, which removes phosphate groups from key metabolic enzymes. This process rapidly changes enzymes' activities within minutes. Through this mechanism, insulin stimulates glycolysis and suppresses gluconeogenesis. Specifically, insulin activated enzymes such as pyruvate kinase and PFK2, which enhances the glycolytic pathway and promotes the breakdown of glucose for energy and storage. At the same time, insulin suppresses the expression of important gluconeogenic enzymes such as by reducing the production of these enzymes, insulin decreases the liver's abilities to synthesize new glucose. Overall, insulin promotes anabolic metabolism, allowing excess glucose to be stored as glycogen. In contrast, glucagon functions primarily during fasting or when blood glucose levels fall. and signals the liver it signals the liver to produce and release glucose into the bloodstream. Glucagon activates a signaling pathway involving cyclic AMP, which in turn activates protein kinesis A. This kinesis phosphorylates several metabolic enzymes, leading to inhibition of glycolysis and stimulating gluconeogenesis. Understanding the regulation of glycolysis and gluconeogenesis is not only important for basic biochemistry, but also has significant clinical relevance. Many diseases involve disruptions in the metabolic pathways. One example is hereditary fructose intolerance, a genetic disorder caused by mutations in the enzyme aldolase B. Normally, aldolase B helps break down fructose 1 phosphate in the liver during fructose metabolism. When enzymes is defective, fructose 1 phosphate accumulates in liver cells. This accumulation interferes with normal glucose metabolism and depletes cellular phosphate and ATP levels. As a result, patients can develop severe hypoglycemia, enlargement of the liver known as hepatomegalia, and metabolic acidosis after consuming foods containing fructose and sucrose. Another clinically important condition is type 2 diabetes. In this disease, tissues become resistant to the effects of insulin, even though blood glucose levels are already elevated, the liver continues to produce glucose through gluconeogenesis. Normally, insulin suppresses the expression of gluconeogenesis enzymes, but in insulin resistance, this suppression becomes impaired. As a result, excessive hepatic glucose production contributes to persistent hypoglycemia. Common medication used to treat type 2 diabetes is metformin. Metformin works primarily by decreasing glucose production in the liver. It activates an important cellular energy sensor called AMP activated protein kinesis or AMPK. The activation of AMPK alters the energy state of the cell and suppresses gluconeogenesis, which helps lower blood glucose levels. Finally, alterations in glycolysis are also seen in cancer metabolism. Many cancer cells exhibit a phenomenon known as the Warburg's effect, where they preferentially use glycolysis to generate energy even when oxygen is available. This process is called anaerobic glycolysis. Although glycolysis produces less ATP than oxidative metabolism, it allows cancer cells to rapidly generate metabolic intermediates needed for growth and cell division. Changes in the regulation of glycolic enzymes contribute to this metabolic adaptation in tumor cells. The regulation of glycolysis and gluconeogenesis is a finely tuned system that operates at multiple levels to maintain glucose homeostasis in the body. Hormones like insulin and glucagon play central roles by triggering covalent modifications of the enzymes and controlling gene expressions, allowing the body to respond appropriately to feed and fasting conditions. Beyond hormonal controls, cells also use intermediate feedback from energy metabolites and pathway intermediates to quickly adjust enzymes activity. This allows metabolic flux to be finely tuned within seconds, ensuring that energy production matches the cellular demands in real time. A deep understanding of these regulatory mechanisms is essential not only for basic biochemistry, but also for clinical medicine. Disruptions in these pathways contribute to metabolic diseases such as diabetes, inherited metabolic disorders, and cancer. By unraveling how these pathways are controlled, researchers can develop targeted therapeutic interventions to treat or manage these conditions more effectively. Attached is the references that I used.